Today we're going to discuss DNA replication. Now I've asked you to put 1 through 6 of the flip boxes onto page 47 and 48 in your interactive notebook. Now we are going to go through these notes, and this first page of notes you won't have in your notebook. It won't be in your flip book. Okay, but you'll have to know this stuff. Also, folks, okay, our objective is for you to be able to fill out a diagram and to explain what is going on during DNA replication. Now, the first thing that we're going to talk about is what DNA replication is. It is the copying of a double-stranded DNA molecule. Now, we know that DNA, from our studies and our talks about the history of DNA, we know DNA is a double helix. So, DNA replication is the copying of that double helix. Each DNA strand holds the same genetic information. So each strand can serve as a template for a new opposite strand. The parent strand is preserved, and the daughter strand is assembled from nucleotides. So, we assemble the daughter strand by reading the parent strand. We call this semi-conservative replication. Now, that's going to be important, folks. Semi-conservative replication. I'll show you an entire slide on this, and you might even draw it somewhere where you got some space on one of these pages, 47 or 48. Okay? The resulting double-stranded DNA molecules are identical. So, if we look, we have an unzipping of the DNA strand as we show it right there. Now, I told you about semi-conservative replication. I have seen this also, folks. Okay? I have seen this on not the end of course, but like the OGT, the Ohio graduation test that we used to take. All right? So, what is semi-conservative replication? We look at the original parent strand, and I am signifying the parent strand in blue, dark blue. What happens is the original parent strand splits and has a light blue daughter strand. So the two parent strands split apart and form daughter strands. Well, since these two strands are identical, we're going to have identical DNA. So these two DNA are genetically identical to that one right there because they just made copies. The dark blue one is the parent, the light blue one is the daughter. Well, once we separate then this parent and this daughter, we still have a dark blue and a light blue, but right here are two daughter strands, aren't there? But nonetheless, are they still identical? Yes, they are still identical. That is what we mean by semi-conservative replication. Now, let's talk a little bit about DNA replication, and you're going to find this is, ladies and gentlemen, your flip books. This is your flip books. So on the very first square under DNA replication, we should have DNA helicase, which is an enzyme. Remember, we can tell enzymes by the ASE ending. DNA helicase, which is an enzyme, unwinds and unzips the double helix. Now, if you look at what is under that flip book right there, what is there? Nothing, is there? Well, you need to not so much copy this, but you need to draw this picture of the DNA being unzipped, and that yellow triangle there represents what? DNA helicase. All right? So go ahead and copy that picture. If you want to color it, you can color it too. Okay, so the very first step is that the DNA helicase unwinds and unzips the double helix. Step number 2, RNA primase adds short segments of RNA that are called RNA primer. RNA primer. Anybody know what a primer is? Yeah, something that kind of sets up or starts the process, okay, is a primer. So, this RNA primer is an important part. And if you look on your next one, go ahead and color in and label this slide right here. So go ahead, color and label our slide here on the RNA primer and the RNA primase. Now, what I'm going to do here, folks, is I'm going to label a couple of things, okay? First of all, this guy right here, what do you think he is? Okay, what do you think that is? Let me ask you this. What is it doing? It is unzipping. So in this diagram, what is that? That is the DNA helicase. That is the DNA helicase. Something else too that you have an arrow pointing right here, don't you? Don't you? You know what that is? What we call? That is what we call the replication fork, where the DNA strand is split apart. That is what we call the replication fork. Okay, the replication fork.